In this lecture, I'll be talking about a bacterial structure called an endospore, and we're going to describe the structure of an endospore, and as well as talk about how endospores form and how they can turn back into vegetative or dividing cells. And we're also going to discuss why endospores are kind of a particular interest to those in food microbiology as well as in clinical settings. So what is an endospore? An endospore is a thick-walled spore that forms inside of a mother cell. And endospores are considered kind of a dormant structure. Bacteria are not able to divide when they're in an endospore form. And so you can actually see endospores, different types or shapes of endospores. The spores are here in purple, and they form inside of these mother cells in yellow. And endospores are really interesting structures. They're extremely stress resistant. They're resistant to high and low temperatures. They're resistant to drying out or desiccation. They're resistant to UV radiation as well as gamma radiation. And they're also resistant to a lot of chemical disinfectants, including bleach, right? And so an endospore structure from a bacterial standpoint is extremely advantageous because it allows the bacteria to remain dormant, especially through really high stress situations. And so what can actually trigger a bacterial cell to form an endospore and to go into sort of this dormant state? So endospore forming bacteria kind of rotate between vegetative growth or normal cell division and survival as endospores. And so you can see here a vegetative cell would be considered a normal growing, dividing bacterial cell under normal conditions. And generally, in order for a normal vegetative cell to switch to an endospore, it's going to have to be triggered by one of those stressors. And the most common stressor that triggers this transition to endospore formation is a lack of nutrition or nutrient availability for the bacteria. If a bacteria doesn't have enough nutrients or food to divide, it's not going to divide, and it's going to trigger formation of this kind of dormant endospore. And so not all bacteria are able to form endospores. There are only certain species or groups that are able to do it, one of which are members of a phylum called Firmicutes or Firmicutes. You can see one of the members of this group here that's able to form endospores in the genera Bacillus. This bacterium is called B. anthracis, right, which is an anthrax bacterium, or the bacterium that causes anthrax. And there are also members of a genera called Clostridium that are able to make endospores. And two members of this genera here, Clostridium botulinum and Clostridium tetani, cause other illnesses, botulism and tetanus respectively. And so these bacterium, members of the Bacillus and Clostridium genera, are able to form these endospores. And you can see how this could end up being kind of clinically relevant, right? These bacteria are harmful and can cause pretty dramatic illnesses. And the fact that they're able to form these spores means that the bacteria are able to lie dormant in different situations and can ultimately infect us as humans. And I'll talk a little bit about that later. So endospore structure, like what is an endospore made of? We're going to start at the very, very, very deep inside of an endospore and kind of work our way outward. So at the very center of an endospore is this area called the core. And the core is where the DNA and the ribosomes of a bacterial cell would be located. And inside the core of an endospore, the water is removed. So there's a very low water content. There's a very low pH. It's an acidic environment. And there are some molecules, dipicolinic acid, calcium, and small acid-soluble DNA binding proteins, or SASPs, that help keep the DNA that's inside the core from denaturing under any of those extreme stresses that the endospore is resistant to. Moving outward from the core, the next layer is an inner membrane, which provides some barrier to chemical stressors for the endospore. And then the next layer exterior here, you can see in purple, is called the core wall or the germ cell wall. It's made up of peptidoglycan, just like the cell wall of the bacteria. And it provides some additional protection as well. Most of the endospore's volume, half of it, comes from the next layer out, which is in light green, which is called the cortex. And so the cortex is also made up of peptidoglycan. It's not quite as tightly linked or mesh-like as the peptidoglycan in the cell wall, but it does provide an extra layer of protection and most of the thickness of the endospore structure. And then moving outward, there's an outer membrane and then an external coat. And the coat is made up of proteins. And these proteins are really, really bound together or cross-linked, and that provides a lot of additional stress protection. And so you can see that from the core all the way out to the coat, each layer provides some protection and prevention of denaturation of DNA that helps provide that stress resistance to the endospore. External to the coat is a layer called the exosporium. And that exosporium varies from endospore to endospore. It's another layer of protein, and it can actually help adhere these endospores to particular surfaces. And so if you're wondering what these spores look like from a microscopic view, these are all members of the Bacillus genera, and you can actually see the coat labeled in with this blue bracket and the exosporium marked with the red bracket. And you can see the different structures and shapes of these different endospores of these different species of bacteria. And so the whole process of a cell transitioning from vegetative or dividing to an endospore is referred to as sporulation. So endospore formation or sporulation takes about 8 hours. And so normally we would have a dividing cell here, and this cell is vegetative and it's going to transition into an endospore or undergo sporulation. So the first thing that has to happen is these axial filaments are going to form. And what an axial filament is basically an invagination of the plasma membrane of the bacterial cell into the cytoplasm. And then ultimately, these two filaments, this one on the top and this one on the bottom, are going to meet in the middle and form what's called a septum, or basically a division between the two parts of what will ultimately become the endospore. There's a forespore, and then there's going to be the mother cell. So you have invagination of the plasma membrane and then formation of a septum here, making two different structures inside of the bacterial cell. Now, one of those structures, the forespore, or the precursor to the endospore, is going to be engulfed or taken in by this cell here. And you can see that finished process down at the bottom. You've got the forespore completely inside of the cell. And then after that forespore is completely engulfed in the cell, it can start to form the cortex and additional endospore layers. So you can see the cortex forming here, the coat forming here, as well as the exosporium to create that endospore inside of this mother cell. And then ultimately, the mother cell, the endospore will actually leave the mother cell and become a free spore, floating out in the environment in its now stress-resistant capabilities. So here's your dividing cell. There's an invagination of the plasma membrane that leads to the formation of a septum. a forespore, which will eventually become the endospore, and a mother cell. The mother cell engulfs the forespore. You can see that here. And then that forespore basically matures into an endospore. The cortex and coat and exosporium form, and then that mature endospore can be released into the environment. And this whole process, as I said, takes about 8 hours. And so that's great. It's wonderful. That endospore that's now out in the environment is resistant to all different types of stress. But what happens when the stress goes away? That spore is going to want to go back or transition back into becoming a vegetative dividing cell. And so the process of kind of undoing sporulation or becoming a dividing cell again occurs in 3 steps. The first step is activation. Usually this activation is sort of a preparation step for the endospores where they undergo a little bit of heating, and that sort of primes them for the next step in becoming a vegetative cell, which is called germination. There are some small receptors on the inner membrane of the endospore that can detect molecules that are signaling, okay, it's good out here again, there are nutrients available, and you don't have to be a spore, you can actually divide. And so these germinant receptors sense those signals, and they trigger breakdown of the cortex of the spore. They trigger a lot of water to be uptaken or taken up into the core. And the DNA and the proteins in the core can then become functional again. Those proteins can refold. The DNA is released from the complexes that were protecting it so that it can be used to help cells divide. And then ultimately, once that water reaches the core and the enzymes that are inside the core are reactivated, the core can actually burst forth through the spore coat and go out into the environment as a vegetative cell again. And so you can actually see the full process up here. We had a vegetative cell that sensed that there was a lack of nutrients. It underwent sporulation to create a mature spore. And then that mature spore was able to germinate once conditions were good again and become a vegetative cell that's able to divide and live its life relatively easily.